Okay, I will do that now. So can you see my screen?
Yes, we can see your screen, Gonzalo.
Okay, so as we told you before, there are a lot of ways to access to the Flapjack API. There is the graphical user interface that provides a lot of features, but we also have a Python package that provides some extra features, and it's easy to extend. So first, we have here the setup, and here we install the Flapjack package. So I will be running this, and I drop a link to the Colab notebook as well on the chat for you to join me along if you want. So here we first install Flapjack and Loika, for example. So then we set our drive in order to access a folder in Colab. So I will do that. Easy, quick. And here we are. Okay. Here I access to my drive and a drive folder, and I change the folder to Loika, for example, or any other folder that you want. After that, we import all the necessary packages. And the first thing here will, if you're going along with me, it will be to log in. For that, if you don't have an account, this will be a good time to register. So we do the login. I will put my username and my password. Okay. Is someone going along with me, or like to wait him or she to be on the same page? If not, I will following with the explanation. So here we have Flapjack objects. So we import, for example, DNA vectors. We import signals such as CFP, YFP, and RFP. We import media, in this case, simulated media, for example, and a strain, and as well as the biomass signal, which is OD. And we will reproduce some of the graphical user interface workflow. So first, we have the IWBDA a plasmid that we want to analyze, and this is the SBOL diagram of the plasmid. So we have here the promoter J23101 on the 3 positions, controlling the expression of RFP, YFP, and CFP. After that, we have here, okay. After that, we can select a study. In this case, this plasmid is part of the study of context effect. The name of the plasmid is PAAA. We can select the strain, like MG1655Z1, and the media M9 glucose. And we can compare different things here. So the first thing that I want to show you is we will plot the experimental data. And for that, I get the study. Here we have context effect, the vector, PAA. So the things that I stated here before, I made it on the code now. So this is how I call PAAA, MG6055Z1, and the media. After that, we have the same kind of selection that we have on Flapjack here. So we select the study, and this is the study ID, the vector ID, strain ID, and the media ID. Here we normalize by min-max. The subplots are vector, and the markers are signal. And here we plot data. So this is the raw data. We can also change the selection, for example, to plot the mean data. That is what's Carlos shown in the graphical user interface. And we can see here these lines. Anyone is following here to see if they can reproduce this thing of if they want to change the vector. For example, let me see if I can. Flapjack.
Actually, Gonzalo, I have a question about this.
Yes.
So in your plotting functionality, how easy or difficult is it to customize, like these plotting functions? I mean, so let me be a bit more specific. So obviously, right now you've plotted time series measurements for OD, YFP, RFP, and CFP. But let's say you wanted to, let's say you had replicates, for instance, and you wanted to kind of plot them side by side, or so how easy is it to, like, if you had multiple samples and you wanted to kind of overlay them, or have multiple plots, how easy or difficult is it with the library? Like, is there a lot of plumbing, or do you already have some wrappers around the plotting libraries? I'm just kind of curious about that.
I think the easiest way to customize everything in Flapjack is through the pandas data frame. You can download the data frame and then do everything that you want with that in order to plot it properly. I think the features that we have here are a bit constrained to be modifying because of the wrapper that we use on the top of Plotly. But usually you will have a lot of tools to plot what you want to do. And if you cannot do it with the plotting interface of Flapjack, I think the best way to do it is use pandas data frame, and from that plot the desired data.
Gonzalo, may I add something? Yeah. Can you go a little bit up?
Yes.
Yes. So, Prashant, you define the query because you are plotting some data, but you are sending the query inside the plot parameters. So you are sending the study, vector, strain, media. And if you change that query, you're going to get more data. So you could, example, filter for different assays. So you can plot at the same time 2 assays containing the same DNA, etc. So you can do that just by modifying the IDs, actually.
Oh, okay, that's pretty cool. Thank you.
Yeah.
Yeah. So also, if you want to plot the data of PAAA along another plasmid, like with the similar measurements, you can do it adding that, like have the IDs of both vectors on a list. And that will help, like in that case, like plotting 2 plasmids side by side. But in the case, like for individual experiments, like have instead of one, like have a bunch of trajectories, like of each sample. If I understand well the question, that's a bit more tricky, but I think it can be done. And okay, so then we will move to analyze the data. So we see the PAAA raw data. Now we want to calculate the expression rate. So to analyze this, we can compute the expression rate using the direct method on Surkowater et al. 2015. And for that, we import the biomass signal, which is OD. And then on the plot, we have the extra parameter, which is type, that is expression rate direct. We add the insignificant value here, and as well as before, we go ahead and plot it. So one of the interesting features of Flapjack is provide data management and analysis on the same platform. So all this computation is done on the web server. In future, we want to get a better server to have a faster faster loadings of the data and computation in general. But usually this is pretty straightforward. Like the main goal here is kind of standardize the analysis. So if you do the expression rate direct here, or another person make the same thing in another lab, they will get the same results. And here we have the calculated expression rate. But to me, it looks a bit weird that because the expression rate is basically the derivative of our data, right? So for me, this is pretty much constant. So I don't know why I have like a bulk here. And we can use another analysis tool, in this case, the inverse method, which we developed this year in the group, and it used basically inverse problems to characterize the gene expression. And with this, we obtain a more relevant expression rate in this case. And here we see that at the beginning we have a peak on the expression rate, and then it's mostly constant, which is more consistent with like have a peak on expression rate, like changing the derivative here, and then mostly constant over here. Which is more similar here. As well as this plot, and as Carlos shown, we can make a print layout of this print, and it's basically like this. So this is an easy to print layout for your figures if you want to do it in a paper. So this is one format that we said it will be good for publications. So it's easy to make figures, to analyze them, and easy to make figures to make the publication. Then we will go to the characterization, to characterize induction curves. So as we can see here, now we need to select another study. This is the DEDCAS9 inverters. We have some vectors here, and we choose the normal is none, subplot signal, markers are vector, and plot all data points. So this experiment is basically as follows. Here we have IPDG controlling the expression of PLAC that expresses DEDCAS9. And we have the promoter or R0040 that control the expression of a small guide RNA to join to the DEDCAS9, and both suppress the expression of this transcriptional unit, the PA2 promoter that expresses Venus. We also need a transcriptional unit for the repressor. So we have here first J23101 as a kind of radiometric one, so a control of how the system is behaving. And here we express like I and R under the control of J23101 as well, in order to the other promoters to work. So we need the IDs of this, of the study. So we get the study TCAS9 inverters ID, we get the vectors, and then we get the vector IDs, as well as the IPTG ID and the biomes. We plug this into the plotting function. Now, the type is an induction curve, and the analyte is IPTG. The function for this induction curve is the mean expression, and all of the rest is basically the plot options. We have the biomass signal, we don't have normalization, the subplots will be by signal, the markers will be vectors, and it will plot all the data. And here we have the induction curves of this example, so we can see how the CFP behaves along with the OD, because CFP is a control, so it will be following the OD basically, and we have the behavior of YFP, which is under the control of the system. One of the things that we can notice also in the OD is the more you express the Cas9, the more like cells die. So it's a bit bad for the cells to have that amount of Cas9. So here as well, in order to see better the curves, we can change from all data to mean. We'll do that now. Let me see. I don't run one of these here. So I need the objects. Now I can plot it. And as well as the first plot, I can show you how is the layout of like the choose layout for print. This is the one, so hopefully it's useful for the people. I think almost everyone wants customized prints here, but this is a pretty good final result. And here we can see better the curve without all the replicates, just the mean. Okay, any questions so far in this analysis?
I would like to add something. All the figures that you obtain, those are plotly objects, are plotly figure class objects. So you can modify them using plotly.
Okay. Then let me see some chat now. So trying to follow notebook in Colab, what do you expect in continuity drive my drive Loica? Nothing, it is just one folder that I select to have the notebook running. So if you choose any folder on your drive, it will be okay. Even if you don't select one, because it will be the base.
Okay, thanks. I'm on the step where I think I'm supposed to log in. What's the default username and pass? It's doing like giving me a user input box.
Yes, so this is the Flapjack. So when you go to login here, you can register, and that's needed in order to access the API.
Okay. Yeah, got it.
So if
okay.
you can,
yeah, I'm going to the UI now. Yeah.
Okay, I will send this to you now to make it quicker. Okay, here is the link to make an account. Okay, so now I will proceed with the rest of the induction curve. Now, so here we collect all the data that we analyze. And for each vector, for each signal, we have different hill functions or curves, and we wish to parameterize A, B, K, and N, which are the parameters on a hill function. I will show you the hill function. Let me see if we can do it quickly, because this could be a bit abstract if you don't know the function. Go here. Here it is. So A is the basal expression rate, B is the regulated expression rate, K is the input concentration half expression, and N is the cooperativity degree of the input here. So this is what we are parameterizing now. So we do that for each vector and for each signal. We have always as the biomass signal DOD, and here we will parameterize the hill functions that we just see. Here is the equation as well. So here our input is the supplement or chemical IPTG, and the output is the YFP or fluorescence in general. So also all this code is shared with you guys. If you want to play with it, you have the basic structure. You can modify it to do the things that you want to do. And okay, we are ready to go now. We have params here. Let's see. Here are the parameters that we characterize just now. So here's the A for each vector and each signal. So we have for YFP, CFP, and OD, and all the vector combinations. And that's the basic tutorial of Flapjack in order to you to know a basic knowledge and how to use the Python package. So doing a recapitulation here, the basic functioning is you select some Flapjack data objects and import them using the Flapjack packages. So you basically do Flapjack get, and then you select, in this case, a study and the name. And if you want the ID, you can put .id, and here you will have the study ID. Otherwise, you can just get this and then put study and .id, and that could be the input of the function as well. So it's really flexible to you how you want to do the things. But basically, you need the ID of the study here in order to make the plot and the analysis. You have the vector names. If you can provide one vector or a list of vectors, as well as chemical. Here we have the IPTG, a really classic chemical. So the first stage when you're working with this is importing the necessary data objects that you want to visualize or explore. Then we have custom functions in order to plot the data and to analyze the data. In this case, we saw the induction curve analysis. We also saw that we have different expression rate analysis. So we have the expression rate inverse. We have the expression rate direct. And the direct is from Subkow et al. So this is all taken from the bibliography. And there is also the indirect way. So the indirect way is using smoothing filters. Let me see, I think that doesn't work. I don't know why, but we also have the indirect expression rate that is, this doesn't work with anything. I don't know what happened, what I do.
You have no samples.
Maybe it's because I need to run this one.
Yeah.
To import to get the correct study, because then I renamed the study ID. So let me see. Okay, this is the direct. This because I use the same variable to all the different examples. So we delete the previous ones. So here's the direct as well. And I will test now the indirect. See if I can just hold this and test the indirect, which is a smoothing filter, and then derivation of that. So it's based. So what's the problem with the direct expression rate is when you have low ODs, the derivatives can go negative. So it's really sensitive to noise at the first stages of the growing. So that's why we move towards first the direct method, and second to the inverse method when we start to getting weird data of the expression rate. Also, if you want to test this analysis, here is the manuscript. So I will paste that here too. Okay, questions from Alex Fedore. Currently, all of the data is in arbitrary units. Do you have a plan to implement calibration of OD and fluorescence to get cell particle counts and method? Yes, that's a really good question and a really neat thing in synthetic biology overall. This is done in arbitrary units because these are old experiments, but now we're working with the IGM as part of the IGM engineering committee in order to get the correct calibrants. So in future, the data that we will acquire will be calibrated by OD using the microsphere particles or silica particles, if I remember it well. And for fluorescence, we will have methyl. So yeah, following the IGM engineering committee standards for calibration. So far, we have calibrants for GFP, but the engineering committee is working to do calibrants to RGB. So in future, we can have calibrants for that. But for example, this data cannot be calibrated now because there's no calibration for RFP, YFP, and CFP. So will you import calibrated data or include calibration in Flapjack? I think for future versions, we would like to include as part of the controls, the calibrant. So that's part of the kind of the data curation that we will work on future. But yes, that's totally the aim, to include calibration as one step to upload data and to inform, for example, have calibrated data with something that can be distinguishable from non-calibrated data. So we don't want to exclude the people that is working in the arbitrary units, but I think it will be good to let know the people that is working in methyl, for example, and other calibrated units. From Michael Galsicki, yes, that's an image that I have in my Loica folder. So I think that's why you don't have it. It says file not found error. So yes, basically that.
Yeah, it's the image of the circuit. Yeah.
It's just the image. This
Just
image.
the image. Yeah.
Okay. So yeah, good question, Alex. And I think we're on the same page about calibration. It's pretty much needed. And if we want to move to work collaboratively, because one of the major aims of Flapjack is to allow researchers to generate data and work together, because if we advance like separated, we can do less things if we can work all together, and my data is comparable with your data, and they can be compatible and even complementary in order to extract new information. And in future, hopefully people can just access to Flapjack and make paper of analysis on, I don't know, a set of data sets and so on. So that's the goal. The goal is to us to work together. And for that, we need standards. First, the standards and good practices in calibration, SBOL standard, and now hopefully Flapjack helps with the data standard and analysis. Also, different types of analysis can be plugged in easily in Flapjack and can be added. So we are also keen to work with people that has problems and help them to solve them using Flapjack, and then the tools developed in that work will be added to Flapjack. So if you need help to upload data, to have a template for upload your data, we are happy to help you with that. And any question, we're really open, and we want that people use this tool in order to start moving this. Okay, so any more questions? I'm sorry, Michael, for the picture, but it's just the picture. You are not missing any Flapjack capabilities because of that. Yes, so we show the fit of a hill function now, but it can fit any function. And that's the power also of using inverse problems, because we fit to a model. So you have a model that expresses how the data is produced, and then you use that to characterize it. So if you change the model, you can fit to an other arbitrary function. But yes, that's one of the capabilities at the end. I don't know if that solves the question. Okay, great. So anyone's following the tutorial and have any question, or you are in one part that you cannot advance, let me know. I can help you. If you want, we can see the optional task now. In the meantime, so optional utilities here. I will not run this because this is pretty massive computation, but you will have you can do it by your own after the tutorial, and it takes some time because it has a lot of data. So let's see how it works. So first we select the study. This is again the context effect study, and we want the expression rate. So we make an analysis that is mean expression. We select the biomass signal, which is always the OD. Then with that, we make a data frame. And this is the common data frame that you can get from Flapjack. So you have samples, signals, email ID, the selected color, if you choose it, and the expression here because of the analysis. You have time, you have the analysis, the study, the media, strain, vector, supplement, chemical ID, concentration, the row of that sample, the column, so you know the position of your samples. So for example, I notice always that people have problems with the borders of the plate reader of the 96 well plate, so they tend to not use them. I don't know if that's something that just happened to us, but you can discriminate data that is in parts of the plate that you know that it does not behave properly. And you have supplement, chemical ID. I think I go through them. And that's
for that example, the plate edge one. Can you say, how do you address those wells? What do you use within Flapjack to indicate that? Is it like a higher level function, or do you specify all the wells?
Because it is saved when you upload the data, you have the column and the row specified. So you can select here, for example, you can select or remove wells of your essay that will be a 96 well plate. So if your essay is this 96 well plate, you can call that essay like this one and remove the rows and the columns that you know that does not behave well, or you can analyze them like and see the position, for example, plot the position and the fluorescence and see if the fluorescence behave different at different positions of the 96 well plate. I think that's a very interesting feature that it's possible using the Python package.
Something I would like to add. Maybe Michael wasn't when Carlos was presenting how to upload data, but roughly, when you get the file from the plate reader, for example, you fill different sheets. In the media sheet, if you don't want to include that well, you just put none, and that well wouldn't be included. For a 96 well plate, now we're just able to upload 96 well plates, but it's just a matter of a parameter, and that's it.
I think we can recapitulate that after.
Yeah, I will somehow recapitulate everything in the next tutorial.
I think the uploading of data is key because that allows you to use Flapjack.
Yeah.
So we will go through that after the Python package. Okay, so this is more or less a mess. Like, you don't know how to work with it, but pandas have a lot of nice features. So in this case, we can use pivot in order to rearrange this data. So we choose as values expression. The index will be strain and media, and the columns will be vectors. And with that, we can generate a heat map of the data, and this is how it looks. So this is a bit so this is the bigger image, and here we have in the columns, as we select here, we have the index, strain, and media. So MG5065 and top 10, and M9 glucose and glycerol. So 4 rows. And on the columns, we have the gene expression of the different vectors, and the different vectors, and the colors shows the mean gene expression of the system. So here we can see, for example, the difference between top 10 and MG55 on different media. So the gene expression of this strain is less than the top 10 strain, for example, in the case of RFP, mainly on all the different vectors. So we can start to do more in-depth analysis of our data just because we have extra tools due to the pandas data frame. This is how this data frame looks if you don't plot it. It's just the numbers. Then we can integrate SBOL. So we can install PySBOL and use SBOL to query through the URIs and the role. So here we have we set, this is from SBOL 2. So we're upgrading soon to be more integrated to SBOL 3 now, because SBOL 3 allows data, and we want to include data on your designs using Flapjack. So we're working on that as well. And here we set the vectors, and the vectors are referred to as SBOL URI. And for that, we make the document and get the useful information. For example, PAAF does not have SBOL URI. It tells you, so it cannot be included here. And we also see the data frame by the vector. And yeah, that's basically like you can here we add like the RFPTU and YFPTU just because we have access to the SBOL URI. So we have some information, that's a lot of information of the experimental data, and we can get some extra information of the design using the SBOL URI if you want to. Also, we can integrate Loica, which is a Python package for genetic circuit modeling and design. If you don't see the presentation, I will make a brief overview. So Loica is a Python package that connects really well to Flapjack. So let me go to the first one. So this is it. This is Loica. We aim to complete the DBTL cycle and help synthetic biologists to do a more productive work using software tools in general. So our aim is to program biology using DNA. And this is basically Loica. This is very, very simple. It has an operator. It has gene products that could be regulator and reporter, a genetic network, a metabolism, supplement, sample, and assay. And with this, you can generate OD models of the dynamics of your genetic circuit or network. We use genetic network because we think that genetic circuit is a subset of genetic network. So you're more free to use anything here outside of the circuit analogy. So the general mathematical models has a production term, which is phi. This is the gene expression of the operator. We have gamma, that is the degradation rate of the output, and the instantaneous growth rate that dilutes the output or the protein that they're producing. The operator is referred to this part of the transcriptional unit, the promoter upstream element, the promoter and the RBS. So it's the regulator part. It can be a node, a NOR, or a general node. That can be anything. With a gene expression, the gene product is a protein or RNA that the transcriptional unit expresses and is related to the coding sequence. The regulator could be a transcription factor such as that R, and a reporter, which is another gene product, could be a GFP, so it have a signal. The genetic network orchestrates this function and it can generate the OD model, but this doesn't have context, so it's rather plain. So to context, we have the metabolism, and the metabolism can have a synthetic profile based on Gompertz, a growth model, or a characterized profile from data. Here I show the 3 different methods that Flapjack has, but Loica only used the blue one, which is the inverse method because it uses the model in Loica to characterize the method. So this is the only one available direct in Loica. We have a sample that is basically one well of your well plate, which cell, the cell has the genetic network or the DNA in general. It can have supplements like this one, or small chemicals like IPTG. And the assay is the collection of samples. So here we have the whole 96 well plate And Loica can generate synthetic data in this format, so it's compatible with Flapjack, and uploads the data to Flapjack. So this is the mapping of the communication from Loica to Flapjack. Essay, sample, and supplement are the same objects on both. The reporter on Loica is connected to the signal on Flapjack, and finally the genetic network is connected to a vector on Flapjack, which in turn is connected to a SBOL component through SynBioHub URI. And here is the mathematical model of a knot operator. This is basically like the Hill curve of a repressible transcriptional unit. This is the mathematical form. This is how you state that on code. And I will go through the examples more in depth on the same notebook. So now that we know more or less that Loica exists, it is done to simulate and to model and design genetic circuits. First, we import all the Flapjack objects that we will be using, which are these ones: the DNA, the vector, the study, the media, strain, the signals, and the biomass signal. We start creating in this case, the example is a repressilator. So we make a genetic network. We add the regulators, in this case, like I, R, and CI. You can add them separately, or you can add them as a list. You also have the reporter, CFP, YFP, and RFP, and you connect them using operators. Here, we have the functional part, which is a triple knot ring oscillator, as like the repressilators, but this is a balanced one. So this is a NOT gate that has as input the CI and outputs like I, which is the input of the next NOT gate, which outputted R. That is the input of the following NOT gate that outputs CI. That's the input of the first NOT gate, making this NOT ring oscillator. But like I, R, and CI are not measurable. So we need to add measurable signals. So here we have a NOT operator that has as input CI and outputs a fluorescent protein. For all the signals or all the regulators, we provide a reporter to track them. We set the metabolism, in this case, a simulated metabolism using the Gompertz growth model. We set the sample, which has a circuit, the repressilator that we just set here, the metabolism, media, and strain information. Then the assay has the samples. It sets the number of measurements and the interval. So this is like the machine that is taking the measurements of your samples. And you can provide a name and a description, as well as the biomass signal. You run the assay, you obtain this kind of data frame, and then you can upload this to Flapjack. And then use the same Flapjack visualization tools to see how behaves the repressilator. So you can upload to the Flapjack data management tool, and then use that tool to visualize your designs. And as well, I show here that you can have the regular visualization or the publication plots. We can make also a NOR gate here. So we import the DNA NOR and the signal CFP. This is more simple. We just have to instantiate the NOR gate as a genetic network related to a vector. And remember that vector is connected to SBOL URI. We state a reporter, in this case SFP1, that will be a CFP, and we add that to the system, and supplements, in this case HL1 and HL2. So this NOR gate takes as input this HL1 and HL2 and output CFP or SFP1. We set the parameters of the equation. This is basically the same as before, but with this little twist. So we have alpha sub 0, which is the basal expression. alpha sub 1 modulates the expression of the transfer function for the input 1, alpha sub 2 modulates the expression for the transfer function for the input 2, and alpha sub 3 modulates the interaction between both. And that's easily done in this line of code. And then we set up the assay. To this assay, we will add the concentration of the AHL from a log space from -3 to 3. We take 8 samples to fit in the 96 plate reader, and then we upload the data, and we can visualize that on Flapjack. So I have that here. So this is the heat map of this data. So this is synthetic data, but you can do the same with real data. And that's it. I think we go through the whole things. Any questions so far? No? Okay, I think I dropped you the link to the public Flapjack IWBDA. Please feel free to make anything that you want with it. Usually you open it and you can save a copy in Drive, and then it's yours. That's it. Any question, or if you want to me that say again any other thing, or go through one feature once more time, if you are interested, let me know. Otherwise,
yeah. Could you show how to upload that data so that I can get through the thing? If no one else has questions, then
yeah. So is Carlos here? Carlos, do you want to show how to upload data using the front end? Okay, Guille can help as well. Yeah, I think Carlos is not here. So,
oh, sorry, you cannot hear me. Yeah, I can go over it.
Great. We thought you were doing other things.
No, I'm here. Yeah.
Okay.
The only thing.
So the floor is yours.
Okay, let me just open the stuff again. I'm going to turn on my camera as well. So, can you see my screen?
yes.
Yes.
Great. So, to cover how to upload data. Well, first you have to create an account to sign into Flapjack. Either in this case on the web instance, but you can also do this. It's the same procedure if you create a local instance, as Guillermo is going to teach you in the next section. So I log in with my account, then I hover to my username, and then you click upload. And here there's going to be the window to upload the data. In the previous case, we showed data obtained from a plate reader. And it was this file, the one that is provided on the Slack channel. Here, you have to make little adjustments, as I explained earlier. I'm going to cover them quickly to not repeat myself, but this is the file that you're going to get straight from the plate reader. The only thing that you have to change here is the name of the sheet. You have to set it to data, so Flapjack can understand what it's looking for. And then you have to add 4 more sheets. This is also going to be provided on the GitHub as a template for you to be easier to modify the Excel file. Once you have created these sheets, you just have to provide the information in the following format. In the first cell, you have to put the name of the sheet. In this case, it's media, so you have to put media as well. And then on the other rows and other cells of the first row, you have to number them from 1 to 12. And also on the other cells from the H, from the A column, you have to name them from A to H. And then it's pretty intuitive. You just have to put the information for each well that you want to put. Here is something that Guillermo mentioned. If you don't put, if you type here, for instance, none, Flapjack is going to understand that and it's going to discard the information from that well. It's going to understand that it's have not been used. That is different. For instance, in string, we can also type none, but in this case, it's just for you to understand that it's a control, for instance. And also, you can put information for more than one plate in the same field, but you just need to set a blank row separating them.
Mm-hmm.
And also, we have type here none to establish that it's a control. And also chemicals, the concentrations, but you get the idea. It's pretty intuitive to understand, and you will find a template on the GitHub. So after you have modified the Excel file with this information that is needed, you are good to go, and you can upload it to Flapjack. So if...
Sorry, Carlos,
yes?
You said that the link is in the Slack channel. I looked through those links and I'm not sure where.
I'm sorry, I was talking about the zip file that is on the Slack channel. There you can see the Excel that we have provided. I think maybe Guillermo or Gonzalo can help me and put in the chat the link for the GitHub repositories where you can find more information about it.
Yeah, okay, I found the zip file, thanks.
Okay. And after you have the file done, in this case, we have one already provided to you with all the modifications needed, and it's called edited. You can go ahead and select a study from the list if it's from a study that has been already created, or if it's a brand new study, or you can just go ahead and create a new study. In that case, you need to provide a name for the study, a description, and a DOI. This is not mandatory, but it's good if you, for instance, have already published a paper, you can put it here so people can go to it. Also, you can establish here if you want your study to be public or not. You can modify this setting if you change your mind later. So for instance,
could you, before going forward, could you, there's a couple of questions in the chat here.
Oh,
we'll go through it. Yes.
Okay.
So the first is Sai. In the long term, do you envision a library of data analysis presets, etc.? So yes, what I understand of your question is that having different type of analysis and kind of plug them into Flapjack, that's our first idea. Like we aim to that, and we kind of started doing that using the Python package that Gonzalo has just shown. Because you can pull your data and apply custom analysis, but the idea would be to have, as you say, a library of like pluggable modules for Flapjack to perform those analyses. And then, yeah, Alex, the sheet that's called chemicals is for different types of chemicals, including antibiotics. So you can include them in that sheet.
Here, you can specify, and as I said, you just need to put a blank row to separate them, the different chemicals.
Yes, exactly. And all of those chemicals in that sheet, like PTG, ATC, goes on another object, because this object in Flapjack, the chemical object, is just a description of the chemical. So you put that object, the chemical object, into a supplement object, which contains more information regarding the concentration of the chemical, etc. No, it's okay. Maybe you thought that the chemicals were just for inducers or chemicals like that, but it's actually for anything you would like to include. Antibiotics. We were including also cell-free chemicals concentration. So it's pretty flexible in that sense, because then when you go and query your data, you can just filter by antibiotics, for example, for some antibiotic, and then you can see how your data behave depending on changing each of the chemicals in a heal function, for example.
Could you speculate? So let's say that we did want to expand the model overall, right? So we have strains, DNA, chemicals here. Let's say I want to add a different kind of thing, like that's not a chemical. Let's say, okay, so proteins. Okay, so in the assays that we do, we often have very specific proteins that we're testing. So there's a DNA, and then there's a protein that would be separate. What would it take to extend the model to include that?
So you are including DNA in terms of concentration. You are using DNA concentrations. Is that what you?
No. We have DNA is one kind of thing. I'm just speculating that I may need to expand the model overall, and we have DNA, which represents the genes, and maybe but then I actually use a completely separate entity, and I'm just using an example like a protein. Can I expand the model?
You can expand the model, but you have to go into a code. Okay. Yeah, you can. I mean, if you want to extend the model, you need to create new relations, new database tables, models, and then go to the parts in the code where the data is uploaded. So you will get that data and store it into the database, and then modify, maybe adding another endpoints for getting the objects of that table, but it's pretty modular. I will show some of that stuff in the next part. I will go more into the code.
Okay, thanks. Yeah, I'll let you get on with it then. Okay.
Yeah, but that's the idea of well, as a concept for us, and continuing with Sai's question, it's that this should be pre-modular, because right now it's ad hoc anyway for us, or we could get to some researchers, but the idea would be to have these modular features where you add different analysis types or different models for the database, etc. We would like to implement that later using abstract classes or things like that programmatically. For example, if you want to make an experiment of single cell measurements of flow cytometry or whatever, this model is like pre-typed for some experiments, for the plate reader experiments, for other imagery station experiments. But the idea is that to be sufficiently modular and easy to modify. as well. And I also do a lot of work with co-cultures. Can I have multiple strains? Yeah, of course. If you go to a strains sheet.
Just like we have done here with chemicals, you go here to the strain and leave a blank row, and then add them here.
Actually, we do that. I don't know, Carlos, if you have a PAAA spreadsheet.
I don't think so. I have it here.
Okay. Well, in that example, we use different strains. Well, the study was the context effect. So we have different strains in different media with different different DNAs. So actually, it was probably somehow related on what you want to do.
Yeah, but it's following the same structure. So just keep a blank row and then repeat this with the.
No. In the media. You mean to have two strains in the same well, or multiple strains? Yeah, you do what Carlos says.
Okay. So can we continue? After that, you can, as I said, you can create a study. If it's not available, in this case, it is already available, and you just select it from the drop-down menu. We select the type of machine. In this case, the Excel file that we have provided comes from an HDX synergy machine. We select that one, and then we select the file that we have previously edited. And then it's a pretty intuitive process on the front end. You just click next. Now, since we can have more than one essay for a single study, you can name the essay here. For instance, you can say whatever you want to that is more intuitive to know to recognize the essay later. And descriptions. I'm not that creative with descriptions for this tutorial, so sorry about that. This is a description. And the temperature that it was performed. And then you click submit. Now, Flapjack is going to recognize the inserted fields, and we can select them with the one that are corresponding on the database here. It's easier for you in this case, for instance, to just type on the search box and select it. But in the case that it is not already available on the database, it's easy as just clicking create new. You specify the name and an SBOL URI for the case of DNA, for instance. I'm just going to go ahead and type what is required here. It's the same deal for chemicals. If it's not available, you can specify it here. You have to provide a description, and you can, if you want, provide a PubChem ID. And finally, you have to specify the signal. All of this data, remember that Flapjack is matching the one from the Excel to the database. And since this one has been already created, they are available here. But if you want, you can create a new one. You just have to specify the name, the description, and the color that you want the signal to be plotted with. And after that, you just click submit metadata, and it's going to be uploaded to Flapjack. Is that clear?
Yeah, thanks. I followed along and it seems to be loading at about the same speed that yours is. So things are good.
Yeah.
Can I ask you, they're asking about other HDX machines. Yeah, one of the things I was wondering is if you already support a multiple plate read. So we have a stacker for the HDX. So we can do, like a bunch of plates at once.
Yeah, so in the same lines, we can add different machines to this pipeline. So if you're willing to use Flapjack, we can help you to automate this process for your machine.
Yeah, in terms of code, what we will need for featuring new machines would be having the parser. So we have the file from a specific machine, and then we need to write the parser of that information in order to store it into a database. So going again with the modularity in terms of analysis or in terms of modifications of the database for different type of experiments, we would also like to have this modularity in terms of adding different machine parsers. So Alex is saying, yeah, from our Tican Spark, yeah, we can develop the parser for that. Or yeah, we could work together on that.
Yeah, it's a pretty cool piece of software. Thanks a lot.
Yeah, it's as you can see, yeah, it has many features. It's not finished like yet, but what I think is that we made a pretty strong basis, like pretty modular. We use pretty good full stack web development tools, so it's pretty easy, I think. We think to start building upon on top of the existing Flapjack in a modular way.
Yeah, I like the idea that that what you guys provide is that we can install it ourselves. That's very compelling to us. Many pieces of software now are just available for the cloud. But for us, we have to install this kind of thing locally, so that's really good. Unfortunately, at noon, it looks like I'll have to take off, so I won't make the full second part of the tutorial here. But I guess this is recorded, so I look forward to watching it. Yeah,
it's recorded, and then you can send us any question. Sai, so you are able to connect a machine permanently, so that when you run an experiment, maybe overnight, it sends the plate read directly, and a script can run it through the presets you set up to have analysis for you in the morning. This is more this is my answer, and this is more like our aim, like our dream. For example, we are working now with an OpenTrons liquid handler. So, for example, we would like to run some code to Flapjack to be able to somehow modify some of the settings of the OpenTrons, or to controlling the time that experiments start running, etc. So yeah, from the beginning, Flapjack is aimed to connect to either hardware, because we have developed another hardware tool which is called FluoPy. I don't know if you have heard of that, but it's basically a microscope with a Raspberry Pi that performs time-lapse experiments for colonies growing in petri dishes. So we also would like to be able to connect to that machine, because we can control the experiments, we can retrieve the images that the Flowby is obtaining the measurements, so we can start doing online analysis, for example. Yeah, we've also thought on that.
I think you could do that. You would get a lot of industry funding, because last year when I was working on the COVID project, one research team was in UK and one was in China. Mm-hmm. And it's really hard to, it was really like we had to change our sleep schedules so that we could have the turnaround time to have the new primers or designs ready based on the previous test data. And so waiting like for their info is difficult, and Flapjack would be a really good intermediary to make that happen.
Yeah, actually, as Gonzalo replied in the chat, he's got a project of 4.5 lab, which is basically imagine you having a tablet with an application that allows you to connect to different machines or software, and basically controlling your lab by the Internet of Things.
That's more like 5.0. That would be amazing. That's further than what I'm asking. But yes, I think the world would just be Flapjack, and you would just have one giant lab that operates inside of it. But I'm controlling lab from tablet. I didn't think that would happen until I was in my next decade. So it's all ahead of the curve.
Yeah, so we're on the lab. We also develop hardware. So as he told you, we have the Flowby, and we are aiming to either modify an open source plate reader or make a new one based on CNC technologies, like a 3D printer, more or less, connected with ESP32 to provide Internet of Things and Wi-Fi connection to the tools. So that's pretty much our idea, to have Flapjack, the web interface, to control your lab equipment. So the idea on future is to have a tab of, I don't know, lab or equipment, and you can see all the equipment that is connected to the web and the state. It is turned on, turned off, it is generating data. You can pinch here and you will go to the data. It can be being updated on real time. So the machine is getting the data and uploading it to Flapjack, and you can see it on real time, and you can hopefully set a script to analyze the data once completed the experiment. So yeah, we have a lot, we envision a lot of things, but to be honest, we are not too many people on the lab, so we need to focus on some things first, and our developments are rather slow because of that. But we are willing to collaborate with more people in order to advance this technology more. I think that the more, and that's the thing that we want, like work collaboratively with all the people, or the synthetic biologists. We have the same aim, so we can achieve them in less time if we work together. And also that's one of the aims of Flapjack, to provide such a software tool or platform to allow scientists to work together, to share data, to have their analysis tool and share the analysis tools as well. And I made this analysis, you make this, and know that you are using different analysis as well. Like, I get this data using this analysis, and you get this other result on the same data using the other analysis. Let's see what happens. That's more or less the platform that we want to build. And also this is very related to biofoundries. So it could be a tool to handle like mini and collaborative biofoundries. So you can run your BioFoundry here and be connected to a BioFoundry, I don't know, in UK, and you can work together more easily, basically.
Well, I'll see what Chris's lab budget looks like, and if we could get, if we'll get a flow guy, and then Flapjack you at the same time.
Yeah, we are been working with Chris a lot of time because he knows more about SBOL and SynBioHub, and we wanted to be connected to these awesome tools because SBOL has a lot of really cool tools, and I think we're not using them properly. So we want to enhance these tools in order to be well posed for scientists to say, I need to do this because this will make my work way more easier, and I can like to have more features and for researchers to use them, because the more people that use the standard, the more that we can advance together.
So I have a, I guess it would be a suggestion and a question. So for within Flapjack itself, if you were able to create a work list or a sample sheet, which is input to the plate reader, I think that may be helpful, which is in order to kind of tell the software, which is going to write out the result file, which sample goes in which well, being able to map between this software and kind of what it expects to read in afterwards would help that. And then the second question is that I think someone asked about bulk uploads, and I guess more precisely for me, it would be, are you able to do the same kind of reads of data, not from the web interface, but simply from a notebook?
Yes, you can do the same things and more through the notebook. So the user interface is more like the entering to Flapjack, and then more features are provided by the Flapjack Python package that connects to the API. But everything is on the API. So the core is the API. The front end have some features to access that API, and the Python package has the same features and some extra features to do on the API. I think this is also one of the aim, like do a decentralized network of Flapjacks, because I think Guille will touch this theme more deeply on the following session. But if you have a local Flapjack, you can have your own experiments or your data in your machine, but that also can be, if you want, accessible or public to other ones. So there'll be a network of different machines that shares information in a more straightforward manner.
Mm-hmm. Thank you.
Yeah, no problem. Any other doubts, or shall we move to Guille's part? I think we're skilled to go with you, Guille. Any other question about the data uploading that we can resolve now?
Maybe I can just talk a little bit about after you have uploaded your study. Since you are the owner, you have the ability to manage it. And as I said earlier, you can share the study with users that have already created an account on the instance of Flapjack, either your local or the web one. You can make it public or you can delete it. Keep in mind that making public and sharing are separate options. So if you make it public and you have already shared the study with someone, and then you change your mind and make it private again, the person that got access via the share option is still going to have access to this study. And I think that's pretty much it for the upload part through the web interface.